Everyone, my name is Lucas Galdino. This is the work 301 at the Neuroengineering section, presented at the 27th the Brazilian Congress in Biomedical Engineering. So this work is entitled A Single Administration of GBR 12909 Alters Basal Mesocorticolimbic Activity. By definition, a bipolar disorder is a chronic and severe psychiatric disorder characterized by periodic mood swings between depression and mania. This figure here shows the two subtypes of bipolar disorder. Figure A shows the bipolar disorder type 2, by which the patients fluctuate their moods between hypomania episodes and major depressive episodes with mixed episodes as well. And the figure B shows the fluctuation of a mood on the time of a patient which has the bipolar disorder type 1, which is more aggressive because the patient can fluctuate their mood between proper mania episodes and major depressive episodes. These mood stages are followed by different symptomatology in patients. Mania episodes are commonly associated with low need for sleep, increased self-confidence, accelerated thought and fluctuation of ideas, rapid speech, distraction, increased performance of pleasurable activities, and impulsivity, while on the other hand, depressive episodes are followed by anhedonia, severe episodes of sadness, loss of interest in pleasant activities, and evolution. Those symptoms are commonly associated with the activity of the mesocorticolimbic circuitry, which by definition are the junction of the mesolimbic pathway, which is expressed here by this blue arrow, and the mesocortical pathway, which is expressed here by the projection from the ventral tegmental area to the prefrontal cortex in this purple arrow. This circuitry directly modulated by the concentration of dopamine in the system. And by dopamine, we believe that bipolar disorder mood swings are associated with the dopamine concentration in the synaptic cleft, modulated by the concentration of the dopamine transporter in the presynaptic neuron, in a way that elevated concentrations of that here increases the reuptake of the dopamine from to the presynaptic neuron, decreasing the dopamine here and emerging the depressive episodes, and decreasing in the concentration of that increases the dopamine concentration in the synaptic cleft and can emerge the manic episodes. Here, this figure, we can see the 3D structure of the dopamine transporter. In this work, we used chemistry composite, which is the GBR 12909, which is a selective dopamine transporter inhibitor. Here in this figure, you can see a healthy synaptic cleft with that working well. And in the figure B, we can see the data blocked by the action of GBR 12909, which increased the dopamine concentration in the synaptic cleft. And we believe that this is the microenvironment observed in patients in the manic episodes of the bipolar disorder. So GBR 12909 causes a transient increase of dopamine within the mesocorticolimbic circuitry. Mesocorticolimbic local field potential activity of a single dose of GBR 12909 administration was not described so far, and the aim of this work is to describe the basal mesocorticolimbic electrophysiological activity associated with the GBR 12909 administration. So to do that, we used 12 mice divided in two groups, GBR group with 7 animals and saline group with 5 animals. Here you can see in the figure A a scheme of the electrode implants within in the 5 regions of the mesocorticolimbic circuitry: the prefrontal cortex, nucleus accumbens, basolateral amygdala, ventral tegmental area, and ventral hippocampus. Here in this table, we can see the stereotoxic coordinates of those areas we've implanted here. The figure B and C shows the raw signal of these five regions recorded simultaneously, and the future rate data of the same data here. And in the figure D, we can see the animal while we're recording the local field potential data. To do that, we've used a multi-channel acquisition processor from the Plexon with 1 kilohertz of the sample rate. We also filtered the data with using a bandpass filter between 0.5 and 400 hertz. And after several steps of preprocessing and processing data, uh, we've uh, we've described the power spectrum density by using pValve function on the MATLAB. And we describe the theta, beta, low gamma, and high gamma activity of each of the five areas we've recorded simultaneously on these mice. Our first result is regarding the GBR 12909 modulation of the power spectrum density in the prelimbic cortex. We found that GBR 12909 modulates the activity of the low gamma activity in the GBR animals, increasing the power spectrum density of this frequency range. Our second result is regarding the modulation of GBR in the nuclear accumbens power spectrum density. We found that GBR modulates the theta power spectrum density and high gamma activity, as you can see here. And ventral hippocampus, we found modulation in almost all frequency intervals we've described. And GBR modulated in detail the activity of the theta frequency, the beta frequency, and the high gamma activity. In this study, we investigated how increases in dopamine neurotransmission due to GBR 12909 administration modulate the oscillatory power of mesocorticolimbic structure. We found specific oscillatory power changes in three of the recording regions, which is prelimbic cortex, nucleus accumbens, and ventral hippocampus, as I described it before in the results. Our work is the first study to describe the effects of GBR 12909 induced hypodopaminergia and then the electrophysiologic activity of severe mesocorticolimbic regions recorded at the same time. And these results represent a step toward understanding how dopaminergic imbalance affects the functioning of mesocorticolimbic activity, and future studies can build on this work to elucidate how these oscillatory power changes relate to manic-like behaviors. Thank you for watching my talk.